This is Danielle Walker doing lab number 6 using buffers. Introduction. Buffers have an incredible ability to absorb acids and bases in order to keep the solution's pH from changing. This experiment allows students to discover a buffer's activity and ability to be unchanging when exposed to varying pH levels as a result of acid and base introduction. It will also require the use of titration techniques in order to define the concentration of acetic acid. The hypothesis is that the buffer will be able to maintain its stability up to a certain level of acid and base introduction, and the buffer will be able to maintain its neutrality, but will eventually be overcome by the changing pH levels of the various solutions. Also, the buffer may be more effective in certain solutions. The experiment requires the use of household white vinegar, white paper, distilled water, scissors, a well plate, hydrochloric acid, sodium hydroxide, and pH test strips. The first step is to place 2 milliliters of white vinegar into wells A1, A6, B1, and B6. Then add 4 drops of NaOH to each well and then stir. Next, dilute 6, is that moles?
Yes.
Moles hydrochloric acid to 0.1 moles hydrochloric acid through the addition of distilled water to this per instructions to the NaOH as well. Lastly, for preparation, cut the test strips. Follow instructions as described in Part B and C and record observations and data. The test strips will be used per instruction in Part C of this lengthy experiment. Data and analysis. After adding 5 drops of NaOH into each well, the solutions all turn bright pink. And after we stirred the mixtures, the colors remain the same. Then the hydrochloric acid is added to the first well, making the pink color disappear. Next, we add additional chemicals into well number 2. And after 8 drops, the solution is completely lacking in color. Conclusion. It is evident after performing this experiment that once a buffer is introduced to a solution that may either be higher in acidic, may be either highly acidic or basic, buffers quickly and effectively work to rebalance the solution. This is made clear by the color changes associated with the lack of and the addition of a buffer solution. Practical application. Buffers are obviously found naturally in the human body, but they are also used in many areas of daily life. One example of this would be in shampoo. Shampoo contains buffers that balance out the alkalinity of soap that would otherwise cause chemical burns to the user's skin. Buffers.
Vagina.
Oh. Buffers are also used in diaper rash cream. This baby cream is designed with a pH that is a bit acidic, which slows the growth of bacteria, which could cause rash or infection in that area. Are you videoing me?
I did.
Why?
How
Why?
much time do you have left?
4 minutes?
That's
I mean,
gonna
2
take
minutes. 2. To talk slower. The objectives of our experiment.
30 seconds. Has to be 5 minutes.
Oh, okay. To develop familiarity with the concepts and techniques of titration. To determine the concentration of an acetic acid solution. To determine that the acetic acid acetate ion pair can be used as a suitable buffer. And to observe how well a buffer can withstand changes in pH when diluted and concentrated acids and bases are added to the buffer. Also, to observe the ability of water to act as a buffer.